My name is Germán Nolasco. I'm a biomedical science master's degree student at Juárez Autonomous University of Tabasco. My project is named Clinical Features Correlated with Comethylation Models in Mexican Teenagers Diagnosed with Eating Disorders. Eating disorders are psychiatric disorders characterized by disturbed eating behavior. They have a complex etiology where genetic and environmental factors are interacting. Analyzing disturbed epigenetic interaction through bioinformatic methods could help us to identify the mechanisms involved in its etiology. Environmental effects modify DNA methylation patterns, and DNA methylation forms networks that exert effects in phenotype. Comethylation models analysis could detect small effects of epigenetic interactions, reflecting environmental factors' influence in phenotype when they are correlated with clinical features. The objective was to create comethylation models from children and teenagers' DNA methylation samples with an eating disorder. We evaluate age, gender, body mass index Z-score, psychiatric comorbidities, and eating behaviors from 63 teenagers with eating disorders. Also, we use methylation epic microarray to obtain methylation levels from their DNA. Then, we create comethylation models using a weighted gene correlation network analysis. Finally, we correlate these models with clinical features in other subjects. Also, we can perform enrichment analysis of CpG sites from the comethylation models. This study followed principles from the Declaration of Helsinki. Our subjects had a mean age of 14.5 years with female predominance. We observed two groups of comethylation models given the methylation level from beta values. One group had partially methylated values, turquoise, blue, and purple, and the other group had hypermethylated values, brown, yellow, green, red, black, pink, and magenta. In this heat map, we can see the 7 clinical features and comorbidities correlated with different methylation models. The yellow comethylation model correlates with BMI Z-score, conduct disorder, and psychotic disorder. Meanwhile, purple comethylation model correlates with gender, suicide behavior, and attention deficit hyperactivity disorder. Finally, black model correlates with height. We found significant enrichment in pathways from the genes annotated on turquoise and blue models. These models were enriched for pathways that could be influenced by development status of the sample individuals. Partial methylations of these models suggest transcriptional activation of these pathways. Black model was non-significant enriched in morphine addiction, synaptic bicycle cycle, apoptosis, and mTOR signaling pathway. Meanwhile, yellow model was non-significant enriched in alpha-linolenic acid metabolism, VEGF signaling pathway, and hepatitis C. In conclusion, this data suggests that perturbations in DNA methylation could correlate with somatometric measures and comorbidities in individuals with eating disorders. This research was funded by Gonzalo Arronte Foundation, National Institute of Genomic Medicine, IMEGEN, and National Council of Science and Technology, CONACYT of Mexico. I'm also a CONACYT Fellow. Thank you very much.